a short basic information as an introduction, and then what is epistasis, and then epistasis gene, and then six common types of epistasis with phenotypic ratio and example. Hi, hello everyone, welcome to my YouTube channel. I am Soumya. In this video I will share types of epistasis with phenotypic ratio and example. So, to begin with, we know that genes are sequence of DNA which gives informations to particular cell on what molecules to make and when. However, a single gene can also often come in various forms called alleles. Additionally, a single individual can contain multiple alleles or versions of the same gene. The individual might only express one of the alleles physically or a mixture of the two depending on the type of trait. Phenotype is the physical expression of a gene or a allele, that is called as phenotype. There are two types of traits, that is monogenic trait and then polygenic trait. Monogenetic trait, as in the name indicates, mono means a single gene or a set of alleles determines the phenotype of the individual. And in case of polygenic trait, those are determined by multiple genes or multiple set of alleles. So this is the basic, right? So now if you have to know what is this epistasis, what does epistasis mean for a gene or a trait? Here, coming to the definition first, epistasis in simple term can be defined as the phenomenon of one gene affecting the physical expression of another gene. The word epistasis means to stop or to stand upon. That is the word meaning here. Epistasis occurs between genes of polygenic traits, not on monogenic trait. Okay, it is occurs between the genes of polygenic trait. It is important to note that not all polygenic traits demonstrate epistasis all the time. For a polygenic trait to demonstrate epistasis, there must be an epistatic gene present. So, what is this epistatic gene? Epistatic gene is a gene which, if present, will suppresses or interfere with the effect of another gene. These epistatic genes are also called as inhibiting genes because they inhibit the expression or phenotype of another. So epistasis is therefore caused by the presence of an epistatic gene among other genes which all determine a polygenic trait or phenotype. And there are six common type of epistasis, and I have made the list here. You can have a look here. The first one is dominant epistasis, second one is recessive epistasis, the third one is duplicate recessive epistasis, fourth one duplicate dominant epistasis, fifth one dominant recessive epistasis, sixth one duplicate gene or and cumulative effect. These are the six types of epistasis. I'm not explaining all these six types, but I'm explaining the phenotypic ratio so that you can remember this for a longer time, and you can glance as a quick notes during the examination. So I'm hoping this video will help you to, and it makes you easy to understand the epistasis. Now, one hint, one trick, or one tip that I would like to give here is, which is very important to remember all these phenotypic ratios, because in competitive examinations like KSET and NET, they will ask the phenotypic ratio. So, to remember that, I have a trick here. You can look at the dihybrid cross test cross. We know that Mendelian dihybrid cross test, and the phenotypic ratio is 9 is to 3 is to 3 is to 1. That is the phenotypic dihybrid cross test phenotypic test cross ratio. And here in epistasis, the first one, if you take the first one, dominant epistasis, the ratio is 12 is to 3 is to 1. And so, how to remember this? See here, it is saying in the name indicates it as dominant. So, you can look at there the dominant ratio there, phenotypic ratio. So, as it is a dominant, we have to count the dominant. So, here 9 plus 3 is 12. Okay, so 9 plus 3 is 12. So 12 is to 3 is to 3 is to 1, that is recessive. We should keep 3 is to 1 as usual. And coming to the example here, the alveran color of the barley is the example for dominant epistasis. The next one, recessive epistasis, as in the name indicates recessive. So we have to add the recessive phenotypic ratio here, that is 3 plus 1 is equals to 4. So you can write like this, 9 is to 3 is to 4, and the example for this is coat color in albino series of mice. Albino means it is related to the skin color. Okay, the in human tyrosinase is the one which help in production of that gene actually help in the production of melanin pigment. Okay, if there is a mutation or if there is a absence of that, there is no coloration. So that actually albino means coloring. Okay, so the coat color in albino series of the mice. And the next the third one, duplicate recessive epistasis. Duplicate recessive epistasis, here the ratio is phenotypic ratio is 9 is to 3, 9 is to 7. So here duplicate recessive they are saying. So if you have to you should add the recessive ones here. So 9 you can keep it as usual. So 3 plus 3 plus 1, so 6 plus 1 7. And the next one, duplicate dominant epistasis. Here the duplicate, but here is the dominant one. So dominant one, we have to add the dominant ones here, 9 plus 3 plus 3. So 15 is to 1. And the example for this is feathers on shank of the birds. And the next one, dominant recessive epistasis, and the ratio is 13 is to 1. Here you have to add 9 plus 1 plus 3, so it will become 13 is to 1. Maize kernel color. And the last one, duplicate gene and cumulative effect, that is dominant gene interaction here, that is actually dominant gene interaction here, that is so the phenotypic ratio is 9 is to 6 is to 1. Example for this is fruit shape in cucurbita. So 9 is to 6 is to 1, you have to add the middle one. Okay, so 3 plus 3 6, so that's when 9 is to 6 is to 1. So this how you can remember the phenotypic ratio of the epistasis and of the all 6 types of the epistasis. I'm hoping I have shared the knowledge that I have. I'm hoping that this video will also help you. If you learn something new from this video, please do share and subscribe. Thank you all.